Welcome, folks. So we're going to continue our discussion of microbiology today, but we're going to pivot a little bit. So far in this class, we've talked about microorganisms, the history of microbiology. We've talked about scientific method and tools to explore living systems. We've also talked about cell structures, specifically prokaryotic cells, and the diversity within prokaryotes and specifically bacteria, but we also talked a little bit about archaea as well. Today, we're going to pivot a little bit, and instead of talking about definitely living cells, we're going to talk about what are called acellular agents. And these are potentially pathogenic components, but they are not fully living. They don't have all of the capacity of life. And we've already been introduced to some of these when we had our discussion about viruses. We're going to come back to those. That's going to be the bulk of our discussion today is on viruses. We're also going to talk today about prions, which are proteins that can be infectious as well. So go ahead and pause the video, review any previous content you'd like to review, preview chapter 13, and come back in. Okay, welcome back, folks. So, as we said, so far we've really focused on living systems. What are the components that are needed within a cell for it to be alive, particularly prokaryotic cells, which are very, very simple, but they do have all of the components of life. And we had reviewed those kind of like, what are the characteristics of life earlier in class, and all of those things. So the use of nutrients and energy, the ability to reproduce, the response to stimuli, the organization and kind of homeostasis, the collective evolution over time, all of those components are present within prokaryotic cells, within cells generally. But our discussion today is going to go even beyond that to more simple components, not cellular components, not the cells or prokaryotic cells that have all of the components of life. No, we're going to focus in on viruses, which have some similarities with living systems and can definitely impact living systems, but they themselves are not fully alive, and even prions. And prions are even more unique in that they are so very simple. They're just a protein, but that protein can be infectious. And so that focus or that awareness of what a living system is will help inform us as we're talking about these acellular agents, which are non-living but definitely able to impact living systems because they have similarity to living systems and they have components which can engage living systems and generate pathology. So let's start off with a discussion of viruses. Let's first say, like, how are viruses different from a prokaryotic cell, for example, or a bacterial cell? And then we'll kind of add more and more detail as we go. So viruses, the most obvious initial thing is that they're so small. They're so small. So we were talking about bacteria being between like 1 and 3 micrometers, okay? Whereas viruses are between 20 and 200 nanometers. And so a micrometer is 1,000 times the size of a nanometer. And so when we're talking about a cell, a bacterial cell, we're talking about 1,000 to 3,000 nanometers, right? 1 to 3 micrometers is 1,000 to 3,000 nanometers. And viruses are much smaller than that, 20 to 200 nanometers. The biggest viruses that we know of, megaviruses, which are very, very large compared to the majority of viruses, are 200 nanometers. But most viruses are much smaller than that. So they are very small acellular components. They're very, very small. Well, what's going on in those very small acellular components? They do have genetic information and they do have some of their own proteins, but let's talk about the genetic information first. They have one type of nucleic acid and never more than that. And at first you might be like, okay, what does that even mean? Well, think of a living cell, like a prokaryotic cell. They have DNA, which stores their genetic information, and then they copy that DNA into RNA. right? So they have two different types of genetic material. They have the blueprint in DNA, they have the copies in RNA. They also have the machinery to pull them through all of those steps of the central dogma of biology, right? So we said we go from DNA to RNA to protein. And those steps, we talked about this in chapter 5 and 6, we talked about this kind of throughout the class. Those steps require a significant amount of nutrients, energy transfers, lots of different enzymes, complex metabolic processes. And so even a very simple prokaryotic cell has all the machinery that it needs to move from DNA to RNA to proteins. Whereas viruses, they do have genetic information. They do have nucleic acids, but only one type. And also they don't have the machinery to move between types, which means that they actually have to rely on the host for that machinery. So a virus might have RNA or DNA, but it won't have both. And that RNA or DNA is going to look like, we'll talk about this more in a minute. It could have double-stranded DNA. It could have single-stranded DNA. It could have double-stranded RNA. It could have single-stranded RNA. But it won't have a mix of those different types of nucleic acids. It will just have one type. And it will not have the machinery to move between types of genetic information, will not have the machinery to move from DNA to RNA to protein. It will actually have to rely on the host for that machinery. Okay? We'll talk more about how that looks like in a minute. Since they don't come in with all the components that they need to actually express their genetic information, they are actually only active within living cells. So they're what we call obligate intracellular parasites. They have to be within a cell to be active. Okay, and that's because of what we just talked about before, is that they do have some genetic information that can be expressed and that can be connected with their systems and their functionality, but that can only be done within a living cell because that living cell has the machinery that the virus doesn't have. And so they're only active within living cells. Because they're only active within living cells, when we then think about how to interact with viruses in the lab, we must know that they can only be cultivated in living cells. So we could not cultivate a virus like in the lab. We work with like TSA or MSA or MAC media. All of these are non-living media. They're just nutrient agar that we grow our organisms on. Well, viruses will not grow on those types of media. They have to actually have living cells to grow in, to be functional, and to be able to be, cultured in the lab. And we'll talk a little bit more about that as well. Okay, so let's add even more detail to this discussion of like what viruses are, because now we understand that they are separate from living cells, prokaryotic cells, for example. They're different from them. They're smaller. They have only one type of genetic information rather than multiple types of genetic information. They're only active within a living cell, and within the lab, they can only be cultivated in living cells or living media. Okay? So let's continue to add kind of some detail to that. These very small particles, they cause infections in all types of living cells that we know of. Okay, so they are very specific. Viruses, because they only come in with a couple of tools when they're infecting a host, those tools tend to be very, very specific to one species or even one tissue type within that species. Okay, but there are so many different types of viruses that we know of viruses that can infect any type of cell. So from all different types of eukaryotes, from animals to plants, fungi, protists, so on, to different types of bacteria and archaea. We even know of some viruses that infect other larger viruses, right? So there are so many different types of viruses, even though they're very specific, oftentimes with a couple of exceptions. Oftentimes they're so specific, but there's so many of them that we know of viruses that infect all types of living cells. Okay, their impact, even though they're very small and they're acellular and they're inactive outside of cells, their impact is huge. And we're pretty familiar with this coming out of a recent pandemic. So they themselves are not metabolically active, they do not grow or reproduce, and they do not respond to the environment on their own outside of a host cell. And they absolutely must replicate within a host cell. So a virus will not be able to reproduce. We don't call it reproduction, we will call it replication. And that happens only within a host cell as it's engaged with the machinery of the host cell. What this means too is that viruses will kind of have two functional forms that we want to be thoughtful of. They'll have an extracellular form, which is essentially just their genetic information protected with some proteins, maybe an additional layer. But then once they're inside of the host cell, they absolutely have to uncode and expose that genetic information to the cell's machinery. And so their intracellular form and extracellular forms will be a little bit different so that they can be active within the cell. Okay. Let's go back to this discussion of size. How small are these viruses that are obligate parasites of cells that are not alive or active on their own, but they do have a huge impact on living systems once they engage a host? Well, let's think about the scopes that we've been using in lab. Red blood cell is going to be visible at 400 magnification to 1,000 magnification. We're going to see it as a small circle on the slide, but we're going to be able to see it pretty clearly. Whereas a bacterium, which we've also seen in lab, so here's E. coli, which is about 3 micrometers, maybe 1 to 3 micrometers. And so, in terms of nanometers, that's 1,000 to 3,000 nanometers large. And you guys are familiar with how small E. coli looks like under the scope. Even when we magnify it 1,000 times, those 1 to 3 nanometers, micrometers, or 1,000 to 3,000 nanometers are very, very small to our eye. Okay. Well, the viruses that infect E. coli, they're called bacteriophages, and there are many different types, but viruses that infect E. coli are then 10 to 100 times smaller than those E. coli cells themselves. So very, very small. And most viruses are not going to be visible under a compound light microscope that we use in the lab. We're usually going to have to use an electron microscope to be able to see them. Now, there are a couple of exceptions to this, of very large viruses, megaviruses, that are just visible under a compound light microscope. But generally speaking, they're going to be very, very small. Okay. But they have a huge impact, right? So we can actually sort of think of a virus of a living cell sort of like a computer virus. So a computer virus is essentially lines of code that are similar to the lines of code in the programs that are in our computer systems, but they're just, a smaller or abbreviated package of code. And that code can then engage with different files on our computer or different programs or different apps within our phone and actually impact those apps. And then they can replicate or proliferate through the network as they're engaging those files. Maybe people are sending the files that then have that virus now associated with it. And so they're actually hijacking the normal processes within our files, our apps, our computers, our networks, and allowing that to be the way in which they can impact the system as a whole. And so that kind of analogy of a computer virus, where it's not a whole app or file itself, or it's not a whole app or functional unit itself, but the lines of code or that component is able to impact files, apps, networks, so on and so forth. Okay. So that kind of gives us an idea of what viruses are. We want to break those pieces down. Like, what does a virus look like? How does it infect its host? How is it actually replicating? What are the impacts? And we'll do that in a minute. But what I'd like you to do is pause the video, and then there's just a 1+ page summary of what viruses are from a book that I think is very clear and succinct It's called The Fundamentals of Molecular Virology. And it just is a one-page summary, one and a little bit page summary of what viruses are. So go ahead and jump into that, read through that one-page summary, then come back into the lecture and we'll continue our discussion. Okay, welcome back. So let's talk more about viruses, what they do, what they look like, what are their components, how they engage with the host, and what are the impacts. Okay. So we said that viruses are obligate intracellular parasites. Obligate means required. Intracellular means they have to be inside of a cell, and parasites means that they're engaging with that host in a way that's actually usurping or hijacking components. Okay, and so all viruses have this, like, they are obligate intracellular parasites. We don't know of free living viruses that can just kind of, be active on their own. They have to be obligate intracellular parasites. Now, since they're so simple, they're so small, they're so simple, and we'll talk about some of that simplicity and how small their genome is, they only come in with a few tools. And so viruses tend to be very specific in their hosts. They infect one species, for example, and even within that species, they might infect one type of tissue if it's a macroorganism. There are a couple of exceptions of generalists. So we know of SARS-CoV-2, which is the causative agent of COVID-19. It is more of a generalist. It can infect, bats, deer, humans, a couple other species, but that is unique within the viral world. West Nile virus is another example where it's a little bit more of a generalist. But generally speaking, viruses are very, very specific to a very, very specific host, even a specific tissue. So like various types of viruses that only infect the liver, right? And so, they just tend to not have a lot of machinery. So tools, they have to kind of be very, very specific in what or who they infect. Okay. But as we mentioned, there are so many types of viruses. There's such diversity among viruses that there are types of viruses that infect all living systems that we know of. So here you're looking at a virus that infects tobacco plants. And this is actually one of the first viruses that was kind of discovered as the tobacco mosaic virus. And so essentially, tobacco is a very important kind of commodity, particularly in the 1800s. There's like a lot of kind of focus on tobacco as a commodity. And there were various researchers looking into some of the diseases that were infecting tobacco plants. And so for one of these diseases, there was a hypothesis that it was a bacterial disease, this tobacco mosaic virus, but it was looking different from bacterial diseases, right? And so there were a couple of researchers that were studying this particular disease, and one of these researchers was filtering out the sap from infected plants with a filter that was small enough to catch any bacterial components. Any bacterial components. And the idea was if he filtered out the sap and then took that sap and introduced it into a new undiseased, non-diseased, or healthy host plant, then that plant would not get sick. Right? And so you can think back to Koch's postulates where you're going through and you're trying to kind of select out the agent that might cause the disease, reintroduce it into a healthy host. You're going to need a control for that. And so in this case, he was actually trying to filter out any bacteria and show that if you filter out all bacteria, then a new healthy host would not become infected. But that was not the case. When he filtered out the sap with a filter small enough to catch any bacteria, the disease was still then caused in the new healthy plant. And so he was saying, okay, well, somehow this liquid is infectious. And it was not true that the liquid is infectious. There's actually particles, tobacco mosaic virus particles, that were so small that they were smaller than the filter that he was using, and they were not bacterial. And so there started to be this awareness of these very, very small disease-causing agents. And tobacco mosaic virus is one of the first kind of examples that folks started to kind of understand. Here you're looking at bacteriophages. So bacteria. Phage is in reference to virus. And so whenever you see that suffix phage, you're likely talking about a virus. And so these are viruses that infect bacteria. Bacteriophages often have these really cool structures where they have an icosahedral head, and we'll talk more about what that looks like, but they also have these fibers and tails and receptors that allow them to just directly inject their genetic information into bacteria. And so those are pretty interesting and unique. And we also have some enveloped viruses that are viruses that infect animal cells. And they actually have an envelope, which is this additional layer that kind of looks similar to an animal cell's membrane and allows it to more efficiently engage with that host. Okay. So across the board, there are so many different types of viruses that infect so many different types of organisms. We even mentioned that there are some viruses that are smaller viruses that infect larger viruses. But what's similar between all of them? Let's talk about that. So a virus, if we're just talking about one particle of a virus, we call that a virion. And all virions must have two components. Now, sometimes they have more components, but they must have two components. That's the most basic structure of a virus. Okay, so the virion will have genetic information. So here, some sort of nucleic acid. It might be DNA, RNA, it might be single-stranded or double-stranded, but there's some sort of genetic component, and it's protected by a protein coat, which is called a capsid. Okay, so those two components, you've got to have them for a virus. There has to be genetic information, and there has to be a protein capsid that protects that genetic information when that virion is in the environment. Okay, and so this aggregate of the two parts is called the nucleocapsid. The nucleocapsid, the genetic information just protected by its capsid proteins. Now, a virus that only has those two components is going to have all of the ways in which it associates with its host on its capsid, right? And so like spike proteins on a virus, kind of like the corona, the crown on SARS-CoV-2, those are proteins that make that corona or crown. And those are proteins that allow it to associate and actually attach to receptors on the host cells. And so those, whatever tools that virus needs to infect or associate or attach to its host are going to be on the capsid. But there are some viruses that will actually have an additional layer called an envelope, okay? So the most basic virus is just your nucleocapsid, genetic information with a capsid of proteins around it. But especially viruses that infect animals, they will also have what's called an envelope. That envelope is literally picked up as the virus exits the cell, as it's leaving its host and bursting out of its host. It's literally picked up as part of the membrane of the host, which you can imagine is very, very useful when a virus is trying to evade the immune system, and it actually has an outer coat that's made out of the same components as the cells of that organism, right? And so you could have viruses that have the nucleocapsid and then an envelope surrounding that. These are called enveloped viruses, and viruses that don't have an envelope are called naked viruses, okay? Viruses that have an envelope are going to be more able to engage an animal host because that envelope looks very similar to the membrane of those host cells. But the envelope is actually really easy to degrade. So washing our hands with soap, for example, that soap, that surfactant will very easily break apart that envelope. So enveloped viruses are able to evade the immune system, but they are actually more fragile in the environment. Whereas naked viruses, those capsid proteins are actually pretty hardy against the environment. So they're going to be more likely to be longer lasting on surfaces, fomites, so on and so forth. Whereas your enveloped viruses, less so. And so there's always trade-offs within these different types of kind of cellular and acellular structures, because we talked about similar trade-offs with like gram-positive and gram-negative organisms when we talked about prokaryotes that are actually cellular, right? So you're seeing similar trade-offs here, whereas you could have a naked or an enveloped virus. Either way, Naked viruses, enveloped viruses, they have to have proteins or some sort of way to attach to their host. Those tools will always be on the outermost part of that virus. So enveloped viruses, their proteins, similar to the spike proteins that we just talked about on a naked virus, those that allow them to associate with their host are going to be on the envelope. And they're not going to be on the capsid. And this is really important because if you break apart that envelope, for example, with soap by washing your hands, then this is no longer an infectious agent. If you break apart the capsid protein on a naked virus, which is a little bit more difficult, but it can be done with like bleach, so on and so forth, then it's no longer infectious. But it's that outermost component that has the capacity to interact with the host. And so the envelope on the enveloped virus and the capsid on naked viruses, and not both. Okay. So we said that a virion always has genetic information, capsid proteins, and then maybe optionally an envelope. Okay. And so all of those components, the capsid proteins and the envelope that's optional, those are to protect it within the environment or between infections of different hosts. But the most essential component when it comes to its actual active form within the cell, that's the genetic information. And so that genetic information somehow has to be released into the cell for it to be active. And what that means is if you have a way of preventing the genetic information from interacting with the inside of the host cell, you can also render that virus, non-infectious, right? And so, for example, this bacteriophage here that's injecting its genetic information, if you can stop it from getting that genetic information into the host, then it's no longer infectious. If it's an enveloped virus, maybe you can prevent that envelope from fusing with the host membrane. Then it's no longer infectious. If it's just a virus that's a capsid and it enters into the host, maybe you can prevent that capsid from being uncoded, is what we call, or removed, so that genetic information cannot access the host machinery. Then it's no longer infectious. And so when we get to chapter 10, we'll talk about antimicrobials, which are used against cellular agents, and antivirals. And sometimes these are the tools that we use, is that we actually prevent the genetic information from reaching the host machinery. And that's how we render those viruses non-infectious. Okay, so let's talk a little bit more about that genetic information, or the genome of the virus. Okay, so viral genomes are so very small. They're so very small. Actually, I'm going to move forward a couple of slides and show you how small they are. Okay, so here's an image. Here's an E. coli that's been ruptured, and you'll see that this is all of this is the genetic information of E. coli, and there's probably still more in the cell. Okay, and then this tiny little component here, this is a viral genome that was inside of the E. coli cell. Very, very small. So let's walk through that for a sec. When we're talking about the human genome, we're talking about 20 to 25,000 protein coding genes. So significant amount of protein coding genes. Okay. Now, if we move even smaller to a bacterial genome, something like E. coli, it's just a couple of thousand protein coding genes. Okay, a couple thousand. The smallest and simplest bacteria have just under 1,000 protein coding genes. Viruses, though, usually have literally a handful of protein coding genes, like maybe 9 protein coding genes. The largest viruses that we know of that are exceptions to most of the rules of viruses, they have under 200 protein coding genes. So we're talking about very, very small, very, very simple genomes. And so if a prokaryotic cell, like a bacterium, we said that's like the most basic unit of life, the most basic unit of what a living cell looks like, is a couple thousand protein coding genes. That's how many protein coding genes you need to run all of the functions needed for a living cell. Viruses are much, much fewer than that. literally sometimes just a handful, like, 5 to 15, something like that, protein coding genes. And even the largest ones have so few, just under 200 protein coding genes. So very, very small genomes. What do those genomes look like? Now I'm going to move back 2 slides here. These genomes, they can be, like we said, DNA or RNA, and they can be double-stranded or single-stranded. And this is actually really unique, okay? So let's think back to chapter 7, okay? Chapter 7, we talked about bacterial genomes and bacterial genetics generally. And so we know that within a bacterial cell, we have double-stranded DNA that is then copied into single-stranded RNA. So we have those 2 forms of genetic information, but we don't have other forms of genetic information. There is no single-stranded DNA, and there is no double-stranded RNA. Those are just not present within living cells. But viruses break that rule, okay? So remember, a virus will only have one type of genetic information. We do have viruses that have double-stranded DNA. We do have viruses that have single-stranded RNA. But we also have viruses that have single-stranded DNA, and we have viruses that have double-stranded RNA, which within living systems is unheard of. It's very strange. And so it's interesting that these acellular components, they use the same building blocks that a living system would use, but they use them in a different way, right? They use them in a different way, and that allows them to engage in the very similar molecules that are within a living system, but engage with them in a unique way that allows them to essentially hijack that host machinery. Okay? And so if we go back to the central dogma of biology, which says that we start with DNA, we copy it into RNA, and we make proteins. The end goal is proteins, right? That's the same end goal for a virus. The virus also, its end goal is to be able to make the viral proteins that it needs to protect its genetic information so it can replicate new viruses, right? But since it doesn't have all of the machinery that a living cell has, it will essentially jump into the cellular process at different points depending on what type of virus it is, right? So if it's a double-stranded DNA virus, it's going to migrate to wherever the DNA is in the host. If it's a bacterial host, then it's going to migrate to where, DNA is kind of like concentrated. If it's a eukaryotic host, it's actually going to migrate to the nucleus. And it's going to hijack the process from the very beginning. It's going to say, don't mind me. I'm just a strip of DNA. Yes, you can replicate me and make my proteins. Yay, right? And so it's going to jump in at the very beginning of that central dogma. Whereas maybe it's a single-stranded RNA virus. Okay, a different type of virus that is carrying genetic information that's single-stranded RNA. Well, then that's probably going to jump in right here, right before translation and say, don't mind me, I'm just mRNA, and go into a ribosome, and then you're going to get a protein out of that translation, right? And then you have a couple of unique ones, right? Like retroviruses come in with DNA, like retroviruses, they come in with RNA and then they go back into DNA and then they go through their process. They're very strange, right? And then you have some RNA viruses which come in as a single-stranded positive sense, which means they look just like an mRNA. And you have some RNA viruses that come in as single-stranded negative sense, which means that they actually come in as the opposite of an mRNA, and they have to kind of be converted into an mRNA before they can be then translated into a protein. So essentially, viruses have this diversity of genetic information that they come in with, even though they only come in with one type. But different viruses could have different types. Different viruses could come in with double-stranded DNA or single-stranded DNA or double-stranded RNA or single-stranded RNA, and essentially hijack the host machinery in terms of kind of the central dogma of biology at various parts within this process, right? And that is kind of like the specific superpower of those specific viruses, is that they come in with the tools that they need to then insert themselves kind of slyly in the various parts of the host machinery. Okay. And so even though they have very small genomes, they're able to engage the larger genome of the host and replicate themselves. Well, not replicate themselves, but have the host cell replicate them. Okay. Let's talk a little bit more about what virions look like, and then we'll talk more about replication and how that happens. Okay, so we said that virions, they have genetic information. We talked a little bit about the different types of genetic information that they might have. And then they have capsid proteins around that protects them. Now that protein capsid is made of proteins called capsomeres, and those capsomeres can be arranged in different ways. So just like when we were talking about prokaryotes, there were some prokaryotes that were cocci, some that were bacilli, some that were spirals, just depending on how their cellular envelope and their cell structure was. Well, same with viruses, they can have different arrangement of their capsomere proteins around their genetic information. So you could have a helical arrangement, you could have a polyhedral arrangement, or a complex arrangement. And complex, you can kind of read that as other, because it's not necessarily more complex. Some of them are, some of them aren't. It's kind of like viruses that don't follow the rule in terms of being helical or polyhedral. So here are some examples. Here you're looking at the tobacco mosaic virus, which is just genetic information with those capsomeres wrapped around them in a helical way. So it's kind of like a strand. Ebola is also helical. So here is a student art model of Ebola, and it's just like a long worm-like strand. And so it's folded here, but it could be unfolded where it was just long and stretched out, like the tobacco mosaic virus that you see. And then internal, there is genetic information. So I would absolutely like to thank the student who made that model there. Okay. Then you have your polyhedral. Polyhedral means that it has a geometric faces. So those capsomeres are actually arranged as geometric faces. The most common is an icosahedral arrangement, which is a 20-face structure. And so here's another student artwork. And thank you to the student who made this as well. And you'll have 20 faces, and then you have proteins on the outside of that capsid. Okay, so this would be a naked virus because it doesn't have an envelope. And then inside, internally, you would have the genetic information. Okay, and so that's another example of a virus. And in that case, that is a polyhedral arrangement. And then these other two examples here are enveloped viruses. And these are viruses that on top of whatever the capsid is, and so these attachment proteins wouldn't be there, there is another membrane-like structure, but we don't call it a membrane because it's not active and functional like the membrane of a living cell, right? It's like, but it's made out of the membrane components of the host. We call it an envelope, and it's just the static envelope that has proteins that are usually a couple of viral proteins that are added to it that allow it to adhere to its host and infect its host. And those would be examples of complex shapes. Now, like I said, complex, sort of read that as other, right? Because there are some other shapes as well. So you could have like these bacteriophages, viruses that infect bacteria, where they have an icosahedral head, right? And then they have tails and fibers and various kind of components that can actually attach to receptors on host cells, and then inject their genetic information. So this particular one injects its genetic information into E. coli. Okay, and so the whole virion doesn't actually enter the host, just the genetic information. So in terms of that complex category, so we said helical, polyhedral, and complex, that third category, there's a bunch of diversity in there because it's just kind of like the other group in terms of the structures that virions have and viruses. Okay. So we said that a virus absolutely must have two components, the genetic information and the capsid, that nucleocapsid. But it could also have an envelope. So if it doesn't have an envelope, it's called a naked virus. It's just the nucleocapsid. If it does have an envelope, it's called an enveloped virus. So we want to look at that envelope just a little bit more. Okay. So the envelope, we said, is actually a component of the host membrane. And then what the virus will do is it will actually have some proteins that are viral coded that will add to that membrane. And so the original membrane is a phospholipid bilayer that just pulled right off of the host. But then as it's coming together, it will also incorporate some viral proteins or some viral glycoproteins onto that membrane, and that will allow it to engage with the next host. Okay. This makes it where those enveloped viruses, even though they're more fragile in the environment, this is really important to remember. It's always a trade-off. They're more fragile in the environment because these membranes are easy to degrade. If they dry out, then they're not functional anymore and the virus can't infect its host. If they are broken apart by soap or something like that, bleach soap in the environment, they're no longer able to infect their host. But the advantage is that they can potentially evade the immune system, potentially, although these viral proteins will be red flags to the immune system. And they can very easily engage with the host upon replication and upon infection. So here's replication, where the viral nucleocapsid literally just picks up its envelope from the host membrane. And when it infects its host, we'll see this image later in the slides, it can kind of do the same thing. It can just fuse because its envelope is very similar to the membrane of the host. It can just fuse with the host, and then that lets the nucleocapsid into the host cell. So essentially the reverse of what you see here from 1 to 5. Imagine 5 is number 1. Here's a virus that's actually trying to infect a host, and it comes in and just fuses with the membrane of the host and releases its nucleocapsid into the host. And then the nucleocapsid can be uncoded, and genetic information then is able to engage with the host cell machinery. Okay. So there's a table in your book, table 13.1, that gives you a comparison between what are the properties of a virus and what are the properties of a cell and how those are different. And that allows you to kind of review the content that we've talked about so far in terms of what are viruses, how are they different from cells, what are their components, and how do those components come together to allow them to engage with a host. Now, we want to continue that discussion by talking about what are some different types of viruses very briefly, and then talk about viral replication within a host. Okay, so based on the characteristics that we've already discussed, many of those characteristics are used to classify viruses. So viruses are put into families, even though they're not living, right? They're not alive, they're not living. And so it's not the same type of phylogeny as living organisms, but we do still want to kind of put them into categories called families that allow us to see similarities between similar viruses. Well, what are those families built on? They're built on some of those characteristics that we've already talked about. What is the type of genetic information in that virus? Well, if it's double-stranded DNA, let's look at the other double-stranded DNA viruses and see what are some of the similarities between those. The symmetry of the capsid, right? Is it a helical virus, or is it an icosahedral virus, right? Or is it an enveloped virus? I don't have an example right beside of me of an envelope. Oh, actually I do. Let's look at, I have one over here. So here we go. Here's SARS-CoV-2. This is another student project. And thank you so much to all the students that do these projects. But this is an enveloped virus where that outer component that you're looking at there is actually an envelope. Okay. And so those structural differences also can be used to categorize viruses. What is the size of the virus? Larger virus, smaller viruses, and what are the viral replication strategies, which we'll talk about next. Okay, so here are some viral families. You might be actually familiar with some of these, like Poxviridae. Well, smallpox is in this particular family here. Herpes viridae, papilloma viridae, hepadnaviridae. So you can actually dissect some of these. So like hepadnaviridae. Okay, hepadna is in reference to the liver, or HIPAA is in reference to the liver. DNA is actually DNA. It's actually an acronym embedded into the name, which is sort of funny. So it's a DNA virus that infects the liver, right? And actually, these viruses, like hepatitis viruses, A, B, C, so on, they actually fall into different families. Not all of them are in the same family, but this is particularly hepatitis B. Okay. Some other ones like Picornaviridae. Pico means small. RNA is actually an acronym again, RNA. So it's a very small RNA virus. Here you have another hepatitis virus in this particular group here. And so the hepatitis viruses are not within the same family. They actually fall between different families. Okay, and then you have Coronaviridae, right? Corona referring to that like crown that is very evident and very clear from kind of the structure of that SARS-CoV-2 on the outside there with those spike proteins. Okay. And retroviridae, we said, is a really unique one because it comes in with RNA, then reverse transcriptase is basically allows it to then go back into the DNA structure of the cell, and it hangs out there, but it's actually a little bit active, even though it's hanging out in the DNA. And these are very dangerous. We have a number of cancer-causing retroviruses as well as HIV in this category. So very unique, but also have significant impact. Okay. And so you can see that throughout these different families, we're categorizing those viruses, and you can see some kind of those characteristics that we've talked about. What types of genetic information does it have? Is it large or small? What type of tissue is it actually infecting? And that allows us to classify groups of viruses into similar categories, even though they're not organisms that are alive, and that phylogeny is not the same as like your Linnaeus classification system, but it allows us to kind of categorize. And oh, do we love to categorize as biologists, right? We want to put things as alive or not alive. We want to put them into different, orders and families and classes and so on and so forth. Okay. And so you can see some of that there. Okay. So one of the things that we've seen is really important to viruses, and we've seen it come up in classification, is what is the replication strategy of viruses? And that's something we haven't talked about yet. So let's go into that. Okay. We said that viruses are obligate intracellular parasites. They're not active unless they're inside of a host, and they're not able to replicate unless they're inside of a host, and they are a parasite of that host. They're actually engaging the host machinery in a way that diverts the host machinery from doing what it's supposed to do in terms of supporting the life of that host cell, and it diverts it into the replication of the virus itself. And it's basically that virus is going to use that host machinery because it doesn't come in with those tools. It comes in with one type of genetic information and a couple of proteins. That's it. And those tools have to be enough for it then to sit there as the new ruler of the cell and say, well, I don't have very many tools, but I can tell you what to do, right? And then it will engage as a parasite within that host and force that host to replicate viral components rather than cellular components, okay? But there are two ways that a virus could initially engage a host, Okay, so both of these will include attachment of the virus to the host and release of the genetic information somehow into the host cell. But then what happens next? The end goal is always replication. That virus is eventually going to want to replicate and burst out of the cell. But some viruses will go straight into replication, be like, okay, let's go, right? And they will then go into what's called a lytic cycle, lytic replication cycle. They'll just, insert their genetic information, force the cell to replicate the viral components or the virions, burst from that host, and then start that process over in a new host. Okay. But there are some viruses that will hide first. They will hide within the genetic components of the cell before they go into a lytic stage or lytic phase, lytic replication cycle. You can call it different things, and this is called lysogenic replication or a lysogenic phase. So a lysogenic phase of a phage, right? Because phages are viruses. Okay, so let's look at both of those. So here's like a cartoonized kind of summary of what the lytic cycle looks like, where you have a bacterium that's like, hey, virus, how are you? And then they start to chat, and then the bacterium realizes, uh-oh, that's not a friend, it's a foe. They've released their genetic information into me, and now I am replicating viral particles instead of my own genetic information. And then all of a sudden, there's an aggregation of those viral particles, and they are then released from the cell. The cell lyses, which is kind of to explode, and releases those virions. Okay. At a more scientific level, what do we call these stages? Well, you have 5 stages. You have attachment of the virus to the host. And so that attachment is either going to happen on a naked virus, that's going to be the capsid attaching to receptors on the host. On an enveloped virus, that's going to be the envelope and the proteins on the envelope attaching to receptors on the host. Okay. And then entry looks different depending on the type of virus. So let's look at some options. So you might have a naked virus that engages with the host membrane and just injects its genetic information into the host. Or you might have a virus that the components of the virus, the nucleocapsid as a whole, is released into the host. You might even have parts of the envelope entering into the host, right? And so it just depends on the type of virus that you're talking about. But all in all, there's going to be some sort of attachment with the membrane of the host. And then there's going to be some sort of engagement with the host that allows for the genetic information to enter the host, either with the capsid or without the capsid, maybe with the envelope and the capsid or without the capsid, envelope and capsid, depending on the virus. Okay. Then there is a period that's not on here, which is like an acclimation period, where the viral genome, maybe the capsid is being uncoded, the viral genome is kind of acclimating within the host cell, but pretty soon thereafter, there'll be a synthesis phase, where the genetic information and maybe a couple of proteins, remember that viruses do come in potentially with a couple of proteins or a couple of genes that code for proteins, essentially start to engage the host machinery and force it to replicate the viral genome instead of the host genome. Oftentimes, viruses will actually have proteins that will force the host to actually inactivate its own cellular replication processes and sometimes even degrade its own genetic information, right? Really, really kind of colonizing that host cell. And then once the components have been generated, so you need to have genetic information, you need to have a capsid, and potentially you need to have some proteins that will engage with a membrane if it's an enveloped virus. Those components, once they're synthesized, then they need to be assembled. And so the viral pathogen will actually, or parasite will actually engage with the host machinery to put those components together so virions are actually made and then they can be released. Okay. So here is an example of that process.
Viruses are agents capable of infecting cells and reproducing, but it can be argued whether a virus is alive or just a complex group of molecules. since it requires a host cell in order to reproduce. Whether they are alive or not, they're able to co-opt the host cell's machinery in order to do the bidding of the virus. The replication of the genetic material of the virus, as well as the structural elements that package and protect the genetic materials and enable the virus to infect new host cells. There are many different types of viruses infecting a large range of host cells. They all follow the same general life cycle, with variations in the details of how each accomplishes its replication.
Okay. So here is another example where you're looking at a bacteriophage. So a virus that infects a bacterium, it actually injects its genetic information into the host. The host then replicates all the different components, and that's a significant number of different components that are then replicated. And then they have to be assembled. So synthesized is the correct word. They're synthesized, and then they have to be assembled as part of the replication process. And the host machinery will do that for the virus based on the genetic code that the virus comes in with, that essentially gives the host machinery various tasks, so that this process of replication is completed. Okay. This It depends on the virus, but so some of the most studied bacteriophages, this whole process from attachment of the virus to the host to full-on lysis of the host via replication is about 25 minutes long. So not very long. Now there are viruses that process is a little bit longer and more complex. There are some that are a little bit shorter, but that kind of gives you an idea of how quickly viruses can be replicated within a host once it's been hijacked. And so that final kind of release from the cell, the host cell, that's called burst time. And that gives you an idea of from attachment, which you can see here, there's like a viral attachment, all the way to burst time is about 25 minutes. It enters into the cell. This is that acclimation period that I said is not listed in the 5 steps. Basically, the virus and its components are acclimating to the host, and then you have synthesis and assembly, and then the release of the virions happens usually very quickly because it's literally a lysis of the cell, so it's just exploding outside of the cell. Okay. Here is another kind of more detailed description.
Virulent phages carry out a simple life cycle called the lytic cycle. The phage attaches to the host cell during the attachment stage, and then penetrates the host cell and injects its DNA during the penetration stage. After penetration, the phage causes the host DNA to break into small pieces. The phage then uses the host machinery to synthesize new copies of its DNA. This process is part of the biosynthesis stage. Biosynthesis also involves production of viral proteins. Once biosynthesis is complete, the phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages go on to infect other cells.
Okay. Um, and you can actually see this in the lab if you grow. So what you're looking at here is, for example, a TSA plate, just a plate with media, agar in it. And what's cultured here is not the virus directly, okay? Because we said viruses can't grow on non-living media. What's actually grown on this plate is a slurry of bacteria that have been mixed with viruses. And so you can almost see this is like a negative result where wherever there was a virus, there is a gap in the lawn So the bacteria is not growing in that spot. And that's because those colony forming units have been killed off by a virion and they have been lysed. And so they are not able to grow in that specific spot. And so we call these viral plaques. And this is one method that we can use to grow or enumerate to count the viral load within, an environment or a culture. Okay. And the reason why we have these plaques is because those viruses infect the cell and immediately replicate, burst, and lyse those cells. Okay. So that's one mechanism. And we said that's always the end goal. A virus will always want to eventually move into the lytic cycle. But you do have viruses that are a little bit stealthy, and they can actually first go into what's called the lysogenic phase or cycle. And this is where they infect the host, but they hang out within the host genome. They hang out within the host genome, oftentimes for generations. Right? So the host just replicates. What is the host doing? It's also replicating the genetic information of the virus, right? So instead of 100 virions having to infect 100 hosts, no, one virus or one virion can infect one host, and that host can just continue to replicate. And then all of those daughter cells are carrying the latent phage virus or phage, which is what we call a prophage. Prophage, which is just the genetic information hiding out within the host, which is really cool, but also problematic, right? Because then all of those daughter cells can be induced to then replicate the virus. And that's induction is like some sort of environmental or chemical factor that says, okay, now it's time to come out of hiding and actually force the cell, the host, into the lytic cycle. Okay. And so, interesting fun fact. There are a number of human viruses that can go into the lysogenic cycle and become prophages that our genetic information just carries around. And over evolutionary time, many of these have actually been just incorporated into our genome and have lost the ability to come back out and be lytic. And so that means that in the human genome that we carry in our cells, we're just under 10% viral, which is really cool. Anyways, that is not in the best interest of the virus, though. It does want to be able to be induced into the lytic cycle, which means that that genetic information that's a prophage or just like hiding DNA in the cell's genome does want to be able to come out and actually infect the cell and force it to replicate the virus. Okay. And so this is just kind of a step 1A to the final goal of being lytic. Here is a bacteriophage lambda that infects E. coli. And you can see here that the bacteria can be infected by the phage. And then the phage can just introduce its genetic information into the E. coli genome. It'll become part of the chromosome of E. coli. And then that E. coli will replicate. And then whatever environmental factors induces the word that we use for the switch from lysogenic to lytic cycle, induce it into the lytic cycle, and then it can actually just be replicated and released into the environment and infect new hosts. Okay, and you can see that here.
Temperate bacteriophages carry out two types of life cycle, the lytic cycle and the lysogenic cycle. The lytic cycle for temperate bacteriophages is similar to the lytic cycle for virulent phages. The phage attaches to the host cell during the attachment stage, and then penetrates the host cell and injects its DNA during the penetration stage. In temperate bacteriophages, the phage DNA forms a circle, which can either replicate and be transcribed to produce phage components in the lytic cycle, or can proceed to the lysogenic cycle. During the biosynthesis stage of the lytic cycle, the phage DNA directs the host cell to synthesize viral components. The phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages can go on to infect other cells. Recall that temperate phages can proceed either to the lytic cycle or the lysogenic cycle shortly after penetration. In the lysogenic cycle, the phage DNA integrates within the bacterial chromosome by recombination. The inserted phage DNA is called a prophage. Most of the phage proteins are repressed by two repressor proteins that are products of the phage genes. Whenever the bacterium reproduces, the prophage is also copied. The prophage is excised from the host chromosome in a process called induction. Induction can occur spontaneously through recombination or some other genetic event, or through the action of UV light or certain chemicals. At this point, the phage may enter the lytic cycle.
Okay. And so in that way, we can see kind of those two potential outcomes to a viral infection. Either the virus can go straight into replication and lyse the cell, or it can hang out temporarily within the cell and then go into the lytic phase later on. Okay. And so we're familiar with some of these, like herpes virus, where it can be transferred from person to person, but it can only be, you will actually only see the impacts of that virus under certain conditions, like when the person has a flu or they're immunocompromised or things like that. And that virus is then literally hanging out in the person's skin cells in between those, instances of illness. Okay. Um, so essentially that gives us an idea of what's going on with viral replication. Let's focus in a little bit on the genetic information one more time, just as a reminder. So, that process where the virus attaches and then, engages with the host, synthesizes its components, assembles those components, and then lyses from the cell, it's based on the genetic information, the code within the virus. And that code is what gives the host the instructions or forces the host to do all the various components that allow for that virus to be replicated.